maybe get started this afternoon. I'm Sean Mack. I'm one of the pediatric neurosurgeons. You guys have sat through me talking through the beginning of a lot of these posts. We wanted to highlight some of the research projects that we do in the neurosurgery department here that we've kind of taken you through a big cross-section of neurosurgery in our department over the course of these lectures, starting out with the leadership of both the program directors and the chairman, and then spent some time talking about dynamic personalities in our department and also doing a case conference. And so we've kind of given a big round picture of what goes on here, but the research in advancing neurosurgery is a really important job as well, that even if you're really well trained in neurosurgery, there's a lot of things that neurosurgery as a specialty are lacking and that we all wish we knew more about and all wish that we could do better. And WashU is really well positioned to be able to support research in a lot of different directions. We also have a growing number of research faculty and PhDs who run labs, and Dr. Zipfel has been doing a great job even in a short period of time to ramp those numbers up. But we also have 7 fully funded neurosurgeons who are very clinically active neurosurgeons, but also have ROI funding, which we think is a record. We've been claiming it for a while, and no one's been able to one-up us with that number yet, so we're going to stick to it. And in order to kind of showcase those guys today, we've asked them to all just kind of put together a little short presentation, either speaking extemporaneously just about the research that they do. If you're running a lab, you could talk for 10 hours, let alone just 10 minutes, or giving you a few slides about what are some of the topics that research here and how have we incorporated other people into our lab, or into our, how residents been incorporated into labs.
Yeah, maybe I'll just follow up with, I'll just follow up with that. Welcome everybody. I'm really thrilled to have so many people, 31 right now, on the line. So that's great. I hope these virtual sub-Is have been helpful to some of you to kind of get to know us and our culture and our program. Sean's done a great job of organizing all this. Sophie Church, our coordinator, who's also on the line, has done a great job organizing this. And if you haven't seen it on, we do have a couple of videos that a couple of our outstanding residents put together about our program and about life in St. Louis. So if you have seen those on our website or through social media, I think they're worth taking a look at. They're not too long. If you can't find them, Sophie, I'm sure, can get them to you. But it's just a little bit of a way to get to know us. Like Sean said, I am very confident that we have the most R01 funded neurosurgeons in the country. I've been involved with a couple of kind of review papers through the academy and another mechanism called the K-12 program, which is a neurosurgical career development program that I and Jeff Ojeman and Ahmad Eskandar run nationally. And through those mechanisms, I know how we're doing from that perspective. And that was when we had 6, but we just recruited a new director of functional neurosurgery who is bringing an R01 with him, John Willey. He starts on Tuesday. So I know 7 is the max. So I think we're doing really, really well from that perspective. I think it is the culture of our institution, which is mentoring and nurturing junior investigators up to independent status and beyond. I think our department really embraces that culture that is part of WashU, and that all is kind of a historical credit for that goes to Ralph Dacey, my predecessor, who ran the program, the department for 30 years, and actually the couple of chairs before him as well. So we're really, really proud of it. And I think today we're here to kind of introduce to several of our PIs who are running very significant independent research programs. And I'll have a few slides about my own work and maybe have hopefully some time for discussion or questions at the end. So thanks for making the call.
Okay. We don't have a formal protocol of who's going to go first, but Albert, Kim, you show up on the top left corner of all of my Zoom windows, so I'm going to ask you to go first. So Dr. Kim is a very active neurosurgeon and researcher. He is one of the more popular labs, both for medical students and for residents to join. He's also a really active tumor surgeon, fellowship-trained skull-based surgeon, is very active with starting and advancing our laser program here. I've the skull-based surgeons are sort of in some ways maximally invasive surgeons. He's also worked very hard to pioneer some of the most minimally invasive techniques to be able to reach a tumor. So he'll talk with you a little bit more about the work that he does and how he incorporates training into it.
Great. Thank you very much. Thank you, everybody, for joining us. You know, Dr. Ziffel was saying how we have the most R01 funded investigators as a department. I would say prominently among those investigators are brain tumor surgeons and investigators, for instance, Dr. Gavin Dunn, Dr. Eric Luthart, and myself. But I do want to say that, you know, if you're interested in brain tumors, I'm just making a plug for like a brain tumor neurosurgeon, if that's what you want to be. Across the disciplines, we have many labs that are interested in brain tumor biology. I would say there's something on the order of 11 investigators just specifically interested in brain tumors across you know, multiple departments, and you know, something totaling over $7 million of NIH funding. So if you have an interest, I bet you we have an expert in that interest for brain tumors. Okay, let me share my screen. I just have a few slides. This is really just like a 10,000-foot view. I'll talk about my lab, and we are very much interested in brain tumor cells generally, and more recently we've gotten into the microenvironment. Whoops. And so what I'm showing here in the background is actually a scanning electron micrograph of one of our patient-derived cell lines, actually. This is a cell here, if you can see my arrow, crawling along a fiber made of a polymer and coated with laminin. So it's quite remarkable. These cells, I mean, they look like cancer. They look angry. And in fact, if you give them a microenvironment that looks more like the brain, like axons, like these polymer fibers, they take on very different behaviors and shapes. And one of our hypotheses in the lab is that maybe using these sort of 3D models, 3D infrastructure models might more closely recapitulate what might be going on in the brain. But that's just a picture. But sort of the broad overview of what we do is we're interested in tumor cell state. You know, what kind of tumor cell are you? And there are different axes that you could think about. One that everyone is familiar with is the genetic axis. So there are different subclones or clones within even one tumor, and they're driven primarily by mutations, right? So you can have mutations that define how a tumor cell behaves. And then there's this parallel world, an epigenetic world, really determined by a hierarchy, a developmental hierarchy, where the stem cell, the glioblastoma stem cell, really plays a prominent apical role in this organization of the brain tumor. And we're very interested in both types of organizations, both the genetic and the epigenetic organizations. And so what we do on a daily basis is we use patient tumors, patient-derived tumor cell lines, and now more recently, stem cell-based models to study tumor cell state. And so we can use brain tumor cells, for instance, and make them into primary cells, as you can see here, and then we can do experiments with them both in vitro and when you inject them into mouse brains. Now, a lot of this stuff also involves new omic, sort of genomic, whether it's single cell, and now we're starting to think about and get into spatial new omic platforms, which are really exciting. And all of this work, I have to say, is in close collaboration with buddies of mine like Dr. Dunn, as well as with a new investigator in our department, Dr. Allegra Petty, who is like a world expert in single cell bioinformatics. Somebody's circling my brain tumor there. Okay, so that's a broad overview of the tumor cells and how we're looking at them. We're also interested in the tumor microenvironment, not so much in an immunologic way, like Dr. Dunn, but we're interested in more of the blood-brain barrier, as well as heterotypic interactions, these interactions between the tumor cells and normal cells, like macrophages and microglia, which is a recent interest of ours. But I'll just talk about the blood-brain barrier interest right now, just real briefly. So now everyone's familiar with the laser interstitial thermal therapy. We just did our 300th case recently. This is you know, a great minimally invasive technology that Dr. Luthard basically pioneered back in 2010, and now a number of the surgeons perform this now. But, you know, beyond just killing cells, we don't know that much about it. And so we were interested in what else the laser might do. And so we set up a mouse laser system here with a much smaller laser fiber. And what we did was we would either treat mouse brains, native brain, or implanted brain tumors in mouse brain with laser. And then we would see what would happen. Number one, you can definitely kill tumor cells. That's not a surprise, and that's great. But what we also found was that when you do this for a prolonged period of time, and here is a dye that you can see only going into the parenchyma. This is 3 days, 14 days, and then 30 days after laser. There's this zone of permeability of the blood-brain barrier, which typically is completely closed, as you know, to most medications. And so we think one of the interesting features of using laser therapy might be to open up the blood-brain barrier or the blood-tumor barrier, and that might enable drugs to get in that otherwise never would have gotten in. And this is just a quantitation of that sort of days post-laser. Now, there are other things that might be occurring as a result of laser, and we're also interested in that. We don't know what the cellular milieu as a result of laser therapy is. You know, it might transition it to a state where tumor cells like to grow more or maybe like to grow less, and this is something we're actively interested in. And so just generally speaking, we're interested in ways to study tumor cell state, and with the long-term goal of devising epigenetic therapies against those malignant states. And then we're also interested in how the microenvironmental cells might be affecting tumor growth, and how we could take advantage of technology, for instance, to open up the blood-brain barrier in that microenvironment. And so that, in a nutshell, is sort of what we're doing. I don't know if anyone has any questions about that. Again, it's super collaborative. I would say both projects involve multiple PIs. This one specifically involves Dr. Robin Klein, who's a neurovirologist extraordinaire, as well as Dr. Dunn. So it's a lot of fun just working with my colleagues.
That's great, Albert. I really like your theme of how much you're collaborating with everyone else. And I think that's one of our big advantages here, that we're physically set up, that everything's very centralized. So our labs are nearby, and I think people share a lot because they see each other a lot. Jen Straley is the next person on my screen, so I'll probably ask her to go next if that's okay. Dr. Straley is one of my partners in pediatric neurosurgery. She's a very active neurosurgeon, does everything from fusing spines to taking out tumors. She leads our fetal myelo program that some of you who were tuned in last week may have seen us talking about. Her lab is very interested in the dynamics of CSF flow, that the simple model that CSF gets made in the choroid plexus and it just gets squished out through the ventricles and gets reabsorbed over the arachnoid granulations over the top of the head is probably a gross simplification. And the dynamics of how this fluid gets made and gets reabsorbed is very, very complex, and we don't understand it well at a cellular level or even a molecular level. And some of Dr. Straley's work is helping us to understand that a lot more and have immediate implications in how we're taking care of children and adults with hydrocephalus.
All right. Thanks, Sean, for that introduction. Sorry, I'm having a little bit of, let's see here. Okay, so yes, as Dr. McAvoy mentioned, my lab focuses on cerebrospinal fluid dynamics, and particularly how they relate to disease such as post-hemorrhagic hydrocephalus. And then more recently, we're studying how the tumor CSF ependymal microenvironment relates to outcome in pediatric brain tumors. So I'm just going to share a few slides touching upon my research, just to get an idea of all the different things that we're doing here. Also similar to Albert, my work is very collaborative. Dr. Limbrick studies CSF fluid dynamics as well. And then my research also spans into the clinical realm. And so there's some things that we're doing that really cross the divide between basic science as well as as well as the clinical realm. By way of background, just for myself, I have an MD degree, but spent a few years during neurosurgery residency doing research, and was able to achieve some postdoctorate training to be able to then start a lab. And as you know, WashU is very conducive to building a career as a physician scientist. There's a new, I don't know if it's called a department or a section of physician scientists. So there's a real push to support physician scientists. And I have 3 young kids who may or may not try to Zoom bomb us during this talk. So briefly, these are just a couple examples of CSF pathologies, such as arachnoid cyst in the upper left. You have choroid plexus in the upper right. That's at the sort of scarred choroid plexus after post-hemorrhagic hydrocephalus. The bottom left is arachnoid over area of an intramedullary tumor. And then finally, this is arachnoid, supratentorial arachnoid. And it just shows that there's many different functions of cerebral spinal fluid, as well as the meninges or the arachnoid barriers that envelop the CSF. So there's a lot that we don't know, and it's definitely an emerging field. When I started studying this about, I don't know, 8 to 10 years ago, there wasn't much interest, but over the last 2, 3, 4, 5 years, there's a lot of interest in this. So I'm just gonna skip ahead a few slides here and show you 2 summary slides. So my lab initially started to answer the question, how does hydrocephalus develop after hemorrhage? So as a pediatric neurosurgeon, the most common disease that I treat is post-hemorrhagic hydrocephalus. And we don't have any cure for it or any treatment. So our prenatal babies have bleeding into their brains, and we watch. We watch until they develop hydrocephalus, and then we usually place a shunt. There's another procedure that many of you might be familiar with called an endoscopic third ventriculostomy with choroid plexus cauterization. And a lot of our young infants with PHH do not do well with that procedure. So there's really one option for these patients. So my goal ultimately is to develop preventative treatments for kids with intraventricular hemorrhage such that we are able to prevent hydrocephalus. So my lab uses a rat model of IVH, and these are just a few pictures and examples of, I don't know if you can see my mouse, but larger ventricles after injection of hemoglobin. It turns out of the components within the blood, hemoglobin is most effective at causing hydrocephalus. And I'd also like to mention this, you can see post-hemorrhagic hydrocephalus in cases of subarachnoid hemorrhage and then adult IVH and intracranial hemorrhage as well. So there are further applications to this. We're currently studying treatments with iron chelation. And it turns out that while peripheral administration of iron chelation reduces ventricle size, when you inject it directly into the ventricle, it actually, a single dose prevents ventricular enlargement for up to 2 weeks. So, so we're currently investigating different iron chelators, and really with the hopes of translation to the clinical setting. In terms of a little bit more about the underlying biology, we're really trying to understand the ependymal surface, how it changes in response to hemorrhage, and how these changes can have downstream effects on fluid movement, but also the subjacent subventricular zone, which many of you may know as the source of new neurons. So not only, you know, fluid pathways, fluid flow, but also neurodevelopment. So these are just a few examples in the upper right-hand corner. There's some explants of cilia. So we look at how cilia functions and relate to beat frequency, directional movement, bead tracing studies. And then we use a number of different lines of investigation in terms of methods, as similar to what Dr. Kim mentioned, in terms of SEM, microscopy, confocal microscopy, and then as many of you may know, single cell analysis is really an important tool that we're utilizing as well. The only barrier is that it's at this point still very expensive. So I don't want to take up too much time. I know we probably have a number of other people to go. So although I could talk for a while, I think that sort of sums up most of what we're doing. And please feel free to reach out if you have any questions just about having a, thinking about going into neurosurgery, what type of program is going to be, what you're looking for, and whether or not you're interested in research. I'm happy to speak with you. You don't necessarily need a PhD to start a lab, and our chair, Dr. Ziffel, can speak to that as well. Although it does, of course, help to have that background, you can try to get that experience in other ways. So I'm more than happy to talk with anyone that's sort of on the fence or curious about having a career as a neurosurgeon-scientist.
Thanks, that was really great. We'll go with Gavin Dunn next, if that's okay. Gavin Dunn, you may have seen on one of these lectures before. He's our program director. He's also a very active tumor surgeon, and also has a full lab. So he'll talk a little bit more about the work that he does. And
thanks a lot, Sean. Appreciate that. Is that projecting okay?
Looks great.
Oh, finally, I didn't screw it up. Great. Well, I'm glad everybody is tuning in. You've heard a couple of great talks, and I'm really happy to be a part of this too. I run a lab, as Sean mentioned. I'm also the residency program director, so that's where you may have seen me with Dr. Osmond before. I would echo a lot of the things that that you just heard. The collaborative nature of our community, the clinician scientist phenotype is very much in the DNA of WashU, really throughout all the clinical subspecialties. And I think that it really gets to what the culture of the entire institution is. And so we are not an outlier as a department that is focused on discovery and clinical investigation. That very much is what this institution is all about. And then within the department, you've heard the number of funded investigators and how collaborative that is. And I think I would just crystallize that down to saying that there's a culture of research that is not atypical. And if that is something you're interested, I think you have so many options to find great mentorship and great colleagues. And so I think if you, I think that's a message that is very clear, listening to my other colleagues and what our experiences have been. So my lab focuses really on understanding the immune response to brain tumors. And I have a history in this field. I was an MD PhD student at WashU before, so I've actually had sort of a long view of what this institution is like from student to faculty. And again, it's sort of all the things I was just mentioning. I did my training in Boston and did a postdoc in genomics and functional genomics, and I really kind of bring those two things together in my lab. So I have a joint appointment in neurosurgery and also in the immunotherapy center that is about 6 years old here at WashU as well. So this slide really depicts the central focus of the lab, really trying to understand the immune response to brain tumors, but also within the context of some of the unique anatomy that is basically very specific to the central nervous system and not seen anywhere else. And this is a picture of what I'll call the GBM immunity cycle, and really starting where my mouse is at the top here. You sort of envision that the immune system becomes aware that there's a tumor present. We don't really know where immune responses are primed to brain tumors like we do for tumors and other pathologies outside the brain. We think that antigen-presenting cells may actually make it to the cervical lymph node chains where effector T cells hopefully home back to the tumor and at least recognize, if not constrain, tumor growth. And then also within this microenvironment is the dura with its meningeal lymphatics. And I would say that a lot of the tumor immunology field is kind of focused on some of the things that are wrong or suppressed in this process: the lack of antigen-presenting cells in the tumor, the lack of lymph nodes in the brain, the blood-brain barrier. Our focus has been a little bit different. We just tried to understand how it's supposed to work, and I think that's where the direction of our lab has been. And like anybody studying brain tumors, we hope that our insights into site-specific community lead to something that will help our patients in the long run. So we use preclinical models, patient-derived tissues and cells, and also clinical trial studies to try to address really orthogonally these key questions that I've listed here, really starting at the top. We're trying to understand what are the antigens that in brain tumors the T cells recognize. The second question here is what actually are the antigen presenting cells necessary to generate T cell specific immunity? That has been a long outstanding question in CNS immunology for a long time. 3, how can we target either antigens we identify or neoantigens, which is a class of antigens I'll review in a second. And finally, at a more recent interest of ours is really what is the immunobiology of the dura? I mean, the dura is something we see as cranial neurosurgeons every time we operate, and you sort of get lulled into thinking it's just a structural barrier, but it's turning out to be a very dynamic tissue microenvironment. We've been very interested in studying that. I'll just highlight one of these questions here, which is identifying and trying to target GBM-specific antigens. When these tumor antigens are derived from mutations or DNA structures that are not present in normal tissue, we call those neoantigens. We've spent a lot of time in preclinical models here on the left, trying to identify them, and that process really combines tumor sequencing with prediction programs and also kind of intense immune monitoring methodologies. And I've just showed 2 examples down here of antigens we've identified in a pretty well studied brain tumor line, GL261, and another one more recently, CT2A. And we try to parallel that activity in patients. And on the right is a clinical trial that we have ongoing, where we try to use the exact same pipeline of neoantigen discovery to generate personalized vaccines and what I'll call precision neuroimmunology or precision oncoimmunology. And this is just an example of the immunogenicity of some of the vaccines that we've generated, but really try to study both in preclinical settings and also patients. But studying your patients is something that you really should focus on in your residency if you're interested in research. I think you have a unique opportunity as a neurosurgeon to look at tissue, look at neuroanatomy in ways that other people can't, but it really is an extension of taking care of your patients to study their problems. So I would just sort of summarize our interests really here: antigen recognition, antigen presentation, T cell priming, T cell targeting, and as I said earlier, we're interested in the dura as well, and dural surveillance, and that kind of then leads to what are lists of ongoing projects. I sort of went through a bunch of these. I would just highlight some that I didn't show slides for. Again, understanding ecosystem of human dura, understanding antigen presentation. Recently, we've also become interested in brain metastases, and that's work. Again, both Dr. Straley and Dr. Kim both reference the collaborative nature of a lot of the work we do, and this is a significant collaboration with Dr. Kim as well, looking at brain metastases. And then one project that is, I would say, complementary to everything that we're doing is actually another collaboration with Dr. Chen and Dr. Petty, which is using focused ultrasound to look at liquid biopsies in our brain tumor patients and also to kindle the immune microenvironment. And I bring that up because one of our really great residents, Rupen Desai, is really leading that project. So I'll stop there. That's a summary of our work. But like I would echo the other investigators here, this is a very collaborative environment. People are doing different things, but there are lots of ways that our interests and also technologies intersect. And I think this is a great way really great way to sort of underscore the academic setting that we have here.
Thanks so much.
I'm happy to take any questions too.
I think by my count, we've got one last PI on the line. And I just wanted to emphasize that we kind of group this around people who have R01s, but there are lots and lots of sources of funding and lots of those sources and topics of research that you can pursue as a neurosurgeon and can pursue at WashU. But I think a lot of people work hard to find where they have their comparative advantage and where they find a mentor that keeps them motivated. But people are also funded here through private enterprises, philanthropy, Department of Defense grants. And the range of work is really variable, everything from clinical trials, everything to some of the basic bench tops research that you've been seeing. And one of the themes is people wear a lot of different hats, that you are not only a scientist and not only a physician, but you also are interested in training residents, training medical students, being an administrator to make sure that the environment is there for us to continue to grow clinically and grow in our research. And Greg Zipfel wears multiple hats and looks very good while wearing them. So he's an example of how someone can have 5 different full-time jobs all at once and pull them off very well. So you've seen him talk a little bit in his capacity as the chairman of the program, but now he's going to speak to us a little bit about being a scientist.
Thanks, Sean. Thanks. Hopefully, everybody see that? Okay.
Going around, it looks good.
Is that presenter view or is
Perfect.
that
Perfect?
Okay. So my lab really has, it's really 2 main areas that we work in. Both are R01 funded lab. I have 7 full-time people in my lab with a variety of usually often medical students and a couple of undergrads in the lab as well. And I have 1 neuroanesthesiology attending who's a junior faculty member who's working on getting a K award, who's in my laboratory. But again, there's 2 areas. The first one I'll talk about is the aneurysmal subarachnoid hemorrhage. I'm sure most of you have a good feel for that. The key here is that patients do poorly. I mean, the initial hemorrhage is something we can't do anything about. It's a major driver of outcome. But secondary brain injury related to vasospasm or early brain injury, which is kind of the early development of global cerebral edema within the first couple of days after subarachnoid hemorrhage. Those are 2 big drivers of additional brain injury that impacts outcome. So my lab's very interested in that. It's primarily an in vivo laboratory in terms of how to mimic subarachnoid hemorrhage in a mouse. That's what this picture is here. We have 2 actually subarachnoid hemorrhage models. The one we primarily use is this endovascular perforation model where the suture goes up, perforates the ICA bifurcation, creates a subarachnoid hemorrhage that looks like that. 3 days later, the animals develop vasospasm, as you see here, versus a normal ICA bifurcation. They get short-term neurological deficits. They get cerebral edema, neuroinflammation, neuronal cell death. And it turns out we also start using techniques like optical imaging. And what you see here is a normal functional connectivity from left to right hemisphere. But after subarachnoid hemorrhage, it really disrupts that connectivity from side to side, which we think has a neurovascular coupling underlying mechanism. So those are some of the techniques we use to investigate this. We're trying, my main way, the way I think about subarachnoid hemorrhage and why really we haven't been successful in terms of developing new drugs to prevent or reduce secondary brain injury is that I think we were kind of laser focused on vasospasm or a couple of other entities, and therapies have proven have failed. And I think that a big reason why they have failed is because they're singling out one pathophysiologic mechanism, and it turns out there's a host of things going on. So what we are turning to is trying to tap into endogenous brain protective mechanisms through conditioning paradigms. And the reason that this struck my interest is that one, conditioning and endogenous brain protection can take place within glia, within neurons, and within the cerebrovasculature. So multiple cell types can be protected by conditioning approaches. And there's also a stereotypic window between the hemorrhage and early brain injury, which is about a day or 2, or between subarachnoid hemorrhage and vasospasm and delayed cerebral ischemia, which is 4 to 12 days. And there's a real window there that you should be able to intervene. This is just kind of a real rundown on all the different pathways that are involved with secondary brain injury, as you see listed there. So there's a lot going on. But it turns out there is preclinical and now some clinical data, several papers of which have come out of my lab, as well as this neuroanesthesiology attending that's in my lab. There's clinical data that suggests that there may very well be conditioning effects from ischemia or anesthetics. And if you look at it, almost every entity that's been identified as part of EBI and DCI, there is evidence now, preclinical or clinical or both, that conditioning can reduce or attenuate those entities. So that's why we're excited about it, is because it has such a broad protective effect. I'm not going to bore you and get all the details, but we do have a molecular pathway that we're very interested in for the hypoxic conditioning part of the lab, and it involves NAMPT activation, increased NAD production, which then activates SIRT1, which then leads to DCI protection. And we have a host of pharmacologic genetic data that this is SIRT1 is involved, and that's our current RO1. It turns out we have a host of genetic and pharmacologic data that's implicating NAMPT, and that's hopefully the future RO1. And we're beginning to explore NAD, and in particular with Albert Kim, who is very interested in metabolism from brain tumor perspective. We're going to try to tap into some of his expertise and apply it to this condition as well. So the other area of my lab is really something more chronic, which is vascular contributions to dementia, with a big focus on cerebral amyloid angiopathy. And to the left here is a electron micrograph that shows amyloid, this AMY, that develops in the smooth muscle cell layer, and eventually as it progresses, it can replace smooth muscle cells and even cause endothelial cell dysfunction. This is what it looks like in a mouse model of CAA, and this is kind of a macro view of a mouse model of CAA that we use as a genetically transformed mouse that has a variety of APP mutations that cause it to produce CAA, as well as in some cases neuritic plaques in the brain. The bottom line is CAA causes a host of problems besides hemorrhage. Most neurosurgeons think about it about hemorrhage, and that's the end game. That's kind of when it's most severe and the vessels are degenerating and leading to hemorrhage. But prior to that, it also causes microhemorrhages, but also ischemic stroke. And it turns out that for every one microhemorrhage that a patient has, they probably have 10 microinfarcts. So it's probably a cumulative effect of microinfarcts and microhemorrhage that leads to the progressive dementia that patients with CAA get. But it's probably weighted more towards ischemia than hemorrhage, even though we all think about it from a hemorrhage perspective typically. We have a variety of techniques. It's a mouse model to develop CAA. We use these mice to do cranial window assays like you see here to look at blood vessel function. You can use fluorescent dyes to identify vessels that have the CAA, as you see listed here. And ultimately, we're using a variety of different endpoints and behavioral tests and assays to determine how CAA develops and what we can do to intervene. This is kind of a busy slide from my most recent grant, but the bottom line is we've identified 3 separate molecules, HSPG, NADPH oxidase, and LRP1 that we think are involved with the production of CAA. And it turns out that oxidative stress is at the heart of all this. And so we're using a variety of pharmacologic genetic interventions directed towards oxidative stress to reduce CAA development and improve neurobehavioral outcome in our animals. So with that, I have a nice size lab. Again, it's usually about 7 full-time people with some additional part-time people listed. And we have a great collaborators here at WashU, as well as a particularly strong one at Duke with David Warner and Washing Shen. And what I thought I'd end with is take a step back from my own lab and announce or kind of let everyone know about one really, really important thing that's happening at the institution and for our department that's really going to, I think, just catapult us to another level of research as this happens. So it turns out the School of Medicine is short on a laboratory space, and the dean and the School of Medicine recognized that and had a capital campaign and came up with almost $700 million to build a new research building. And once they had that money and the plan in place, then the idea was, well, who's going to go there? And because of the strengths of neurosciences here at WashU across neurology, neurosurgery, anesthesia, the basics neuroscience department, and others, they decided to make it a neuroscience research building. So there's five different departments who are moving most of their labs, all of their labs, to this new building. This doesn't mean a lot to those who are from WashU, but these acronyms over here are basically our hospital, Children's, Barnes, and our outpatient our outpatient clinic. This yellow is a pathway that's like a tunnel, second floor tunnel that connects this, and they call that the central corridor. And this is the location of this new neuroscience research building, which is a 15-minute walk from the clinical campus. And most of our labs, everybody on this call who've talked about their labs are going to this new building. It's going to be state-of-the-art. There's a brain tumor research center that we're developing in that. Currently, that's 9 investigators with 4 different departments, and we have several additional recruits coming. There's a pediatric neuroscience program, which in totals 9 investigators from 2 departments. 3 of those investigators are from neurosurgery. And there's a cerebrovascular research center with 15 investigators with 6 departments represented. Not all of them are going to the new neuroscience research building, but the preclinical ones are, including my lab and a new recruit that we're working on. And this is just about a 2-minute video of what this thing is going to look like. These are kind of the latest animation of this. It's 2 minutes, just bear with me. Maybe I'll pause it there, but I think you get the feel for it. It really looks cool. And what I don't have to share with you is what the research labs themselves look like. And I told the facilities people, like, you know, all the scientists, they want to see the labs too. And then this is put together mainly for donors, and the donors care less about the labs. So they say they're working on that, and that'll be added to the video eventually. But this is just an example of what, you know, I mean, how, you know, much the department or not how much the institution believes in investing in the neurosciences. And I'll just, one last thing I'll mention is that just to give you another sense on how much they're investing in the neurosciences and neurosurgery. I don't have it on the slide, but if you can see my cursor here, this building right here is a new administrative building. Currently our admin space on the adult side is over here on the corner, but we're moving here in a year to expand because of new recruits. We need new space. And so this is a building that I think was built 5 years ago, 8th floor of the shell space, and they're going to be outfitting it for our new home for our administrative offices. And the reason I picked this, and I'm happy that they're investing in neurosurgery, is that this is, if you look at it, it's right in the interface between our clinical space and our research space. And I think, you know, neurosurgery, probably more than any other specialty, neuroscience-oriented specialty, is really at the interface between the clinical world and the research world. And I think our admin space is going to be at the interface with state-of-the-art clinical and state-of-the-art research facilities for us. So it's really an exciting time for our department, and hopefully you guys are interested in you know, taking a look later with formal interviews and everything. So with that, I'll stop. Thanks.
Thanks so much, Dr. Ziffel. That was great. I haven't seen the video of that building. It looks beautiful. I want to buy a condo in it and live in it. It looks fantastic. I wanted to say, so this is our last one of these lectures, so I wanted to say thank you for the students who are giving us your Sunday afternoons all summer long to come and let us talk to you a little bit. That your generation of neurosurgeons, unfortunately, kind of getting shortchanged on this application session, that you're not going to get to travel around the country in the same way for doing your sub-Is and seeing other places. But we really appreciate your time to consider us, and we hope that we taught you a little bit not only about our program, but about how academic neurosurgery thinks about developing careers from starting residency off to finishing residency and moving on to the next few decades of your career. So we really hope that you think about us when you're sending in your applications, and we're going to think an awful lot about you now and both in 7 years once you're done and are looking for jobs. We're going to be hunting for you again to hopefully join up with us. So just wanted to say thank you for your time and for giving us your attention. Is there any questions?
Anybody have any questions?
Go ahead, Harrison.
Hey, thanks so much for doing this. This is great. I'm curious from the standpoint of, you know, if someone's coming into residency knowing that they're interested in getting involved in a lab and doing some of these projects, potentially looking into R25s and, you know, maybe doing the extended research track. I'm kind of wondering, you know, if they know early on, what does that look like as far as junior residency? Is there any preparation work that they could be doing? I know, you know, junior residency is going to be busy no matter what, but I'm kind of curious, you know, what it sort of looks like leading up to the lab years for somebody who's interested in that.
Gavin, you want to field that?
Yeah, I think, you know, I think that you, it's a great question. And I think that you sort of already said a lot of key elements about the whole process, which is your interest level and, you know, knowing that you're interested in it. I think that finding mentorship and doing these things is just, it's as long as you're interested. There are so many mentors in our department and outside our department, I would also say, but this is a neurosurgery focused thing, so we'll talk about our department. But there are lots of faculty would be more than happy to mentor you, because that's what we're supposed to do. That's our job. And that's part of your training. I mean, we know what that pathway is, and that pathway can, you know, look very different depending on what your interests end up being. But that's our job, is to help you, you know, get the training you need and the expertise and experience you need. If in the long run, being a neurosurgeon investigator is what you want to do. In terms of sort of discrete ways to prepare, again, I think that is something that you really get by just being in our environment and partnering with mentors, because the preparation sort of differs depending on what your area of expertise ultimately is going to be.
And there are several research month blocks in your first several years. So you have time to take those months to get things started, meet with different faculty, and then you're able to carry those projects forward during your more time-intensive clinical months. So, you know, as most of you know, your PGY 2 years is, you know, a pretty busy year, but you have your intern year and then these blocks intermittently to be able to sort of continue forming the background for the R25 or whatever, you know, grant that you would like to apply for. And so most residents, by the time they, you know, hit their research time, they really hit the ground running. It's not that they, you know, arrive on research and then are planning things. So you do have time built in to do that.
That's a great point, Jen. And I'd also kind of two other points I would mention are just one, and Jen alluded to this earlier, there is a division of physician scientists that was recently created at WashU, and that is headed by Wayne Yokoyama, who is a National Academy of Sciences immunologist and also the head of the MD PhD program. You know, their whole mission is to really cultivate and foster the development of clinician scientists, really at a time really in healthcare and educational training where that phenotype has a lot of vectors kind of aimed against it. And part of that is a community of physicians in training called, or CAPSIT, it's kind of a long acronym, but there are seminars and courses and opportunities to study unique patients. And then there's a Dean Scholars program at the conclusion of that if your job opportunity aligns with that. So there's again a tidal wave of interest in preserving and growing the physician-scientist phenotype. But back just to your specific question, Sophie pointed this out on the text that there really is a very broad range, and actually Dr. Straley also alluded to this. There's a very broad range of what your experience level is when you come into residency. Some people like Carl Hacker, who is an MD PhD trained resident in our program. I mean, he was laser focused on a project that he was very interested in, that it really had its roots in his PhD training here, but that then kind of accelerated his ability to not only be our first investigator track resident, but also write for NIH F32, which he has already submitted. So that's an example of someone who sort of is a very kind of self-determined, self-directed person who just needs a slight course correction here and there, but mentorship as well. But he really knew what he was doing. But then then there are folks, and again we have both Dr. Straley and Dr. Ziffel are MD, not PhD, but R01 funded investigators, and that pathway is totally wide open and does not require a PhD. So that just means finding something that you're really passionate about and interested in, partnering with the appropriate mentors here, taking advantage, as Dr. Straley pointed out, of the research months that we have, and also the protected time in the lab when you do go to your research time. And but that's why I said that word culture earlier. I mean, it's not an atypical thing to want to come in here without a PhD, regardless of your prior experience, and want to make research not just a part of your training, but a part of your career and your life. I mean, it's a very normal thing to do, because a lot of people in our department do that. Studying our patients, I think, is part of what we do in neurosurgery and outside of neurosurgery at WashU. So I'm giving you a very long answer to your question, but it's a great question, and I really think it just starts with your interest and commitment, and then it's our responsibility as faculty in our department, our program, to really help you develop that.
Let me just add two things. I know we're kind of telling you and showing you all the different neurosurgery-oriented research experience, PIs, and we haven't even mentioned the PhD scientists that we have, which is a growing group. But I just want to emphasize that we are most interested in you finding the best research program and PI possible. And if that means going outside of neurosurgery for your research time in terms of who your mentor is going to be and your program is going to be, we are all for that. And we have lots of examples of people going into radiology or neurology, developmental biology, neuroscience, genetics, that kind of thing. So we really very much are interested in getting you to whatever it is that's going to be most conducive to you reaching your long-term academic goals. And the other thing is, it's not all about basic science. A fair amount of what we talked about today is about more basic, fundamental discovery type of work. But we are really interested and motivated and have a lot of active programs, including a lot of people who are, running clinical trials and things of that nature. So we want people who are curious, and whether it's basic, translational, clinical, implementation science, what have you. We just want people who want to make an impact and want to contribute and try to make a difference. And what type of research or the style of research really doesn't matter to us, just people who are really inquisitive and want to make a contribution. So,
just had a few general comments. I completely agree with what everyone said so far. I did want to emphasize that you should pursue whatever really excites you. It's not all just discipline and tedium. You should really pick something that you're passionate about and that gives you joy, honestly, is the first thing I would say. in terms of picking your research interest. But then, you know, maybe part of your question is sort of what are the keys to success? Maybe two general things I might say is early identification of the mentor, just as Dr. Dunn and others mentioned. I think that's super important. And then, you know, as early as possible, just trying to have some form of what you want to be at the end of the tunnel. You know, I don't know if it's a lab. I don't know if it's being in a lab, but in a larger lab that's already established. I don't know if you want to be a trialist. I don't know if you want to do clinical outcomes. But if you know the form of it, you kind of want to backfill and reverse engineer how you would want to become that.
Does anyone else have a question? I know we kind of all jumped on and gave Harrison probably 10 times more of an answer than he was maybe even hoping for, but that's kind of how we work around here sometimes.
That was an 11-minute answer to your question.
That was great. Thank you so much.
Any other questions from the group?
Okay, great. Well, thanks again, guys. Really appreciate you giving us your time.
Thanks, everyone. Have a good day.